Hello everyone. So today we will study about the unit 1 of QCHS. In this the basic test for pharmaceutical substances. The first one is amikacin sulfate. Description: it is a white to yellow whitish crystalline powder which is almost odorless. The melting point is 204 degrees Celsius. Procedure: we have to dissolve 10 mg in 1 ml of the water. To that we will add the 1 ml of the sodium hydroxide TS, mix and add 2 ml cobalt 2 nitrate TS. It will produce violet color. Next is we have to dissolve 0.05 gram in 3 ml of the water. Add slowly 4 ml anthron TS. Bluish violet color is produced. Dissolve 20 mg in 1 ml water. We have to add 1 ml barium chloride TS. The white PPT is produced, which is practically insoluble in hydrochloride TS. Degradation test: discoloration of test substances. Second is bacitracin zinc test. In this it is a white or pale brownish yellow powder, odorless with a faint characteristic odor, hygroscopic. Melting point is 250 degrees Celsius. Test is: shake 5 mg with 1 ml of the water. To that add 1 ml of triketohydrin or butanol TS and 0.5 ml pyridine. Heat it to 100 degrees Celsius for 5 minutes. It will produce the white color. Then transfer 0.5 gram to silica crucible and ignite. Dissolve the residue in 5 ml of sulfuric acid TS and filter, and divide the filtrate into the two volume. In the first volume we have to add potassium ferrocyanate TS. It will produce white PPT which is insoluble in hydrochloride TS. To the another filtrate we have to add 1 drop of the copper sulfate 2 TS and 1 ml ammonium mercuric thiocyanate TS. It will produce violet PPT. Degradation test: discoloration of the test substance and non-compliance with following test usually indicate gross degradation. Dissolve 0.10 gram in 100 ml water. It shows the clear, colorless or slightly yellowish solution is produced. Third is the captopril, a white or almost white crystalline powder, odor characteristic but faint. 108 degrees Celsius is the melting point. We have to dissolve 10 mg in 2 ml hydrochloride Vs. To that we will add 1 ml iodine. Iodine color disappear immediately and produces a white turbid solution. Then next test is dissolve 10 mg in 2 ml water. To that add 0.5 ml lead acetate TS. White PPT is produced. Next is dissolve 10 mg in 5 ml ethanol TS. To that add the 0.5 ml tetramethyl ammonium hydroxide or ethanol TS and shake, and again add the 0.5 and add 0.5 ml triphenyl tetrazolium chloride or ethanol TS and shake. It will produce the red color. Fourth is the cisplatin. It is a toxic. Then it is white to yellowish crystal or yellow powder. The melting point is 270 degrees Celsius. In this 5 ml HCl TS is heated to boiling, and to that half solution keep unused. Okay, half we will use half solution first, and half solution is used for the test too. A few crystals of potassium iodide, brownish yellow color is produced, which changes reddish brown on standing. To the remaining test we have to add few crystal of the tin 2 chloride. Reddish orange color is produced, and reddish brown on standing. Transfer: next is transfer 0.05 gram glass dish Add 2 ml sodium hydroxide TS. Evaporate to dryness and dissolve residue in mixture of 0.5 ml nitric acid TS and 1.5 ml HCl TS. Again evaporate to dryness. Orange color residue is produced. Dissolve the residue in 0.5 ml water and 0.5 ml of ammonium chloride TS. A yellow crystalline PPT is produced. Degradation test, discoloration and a change in physical state of substance usually indicate gross degradation. Fifth is the doxorubicin hydrochloride. It is a red-orange crystalline powder, hygroscopic. The melting point is 216 degrees Celsius. Procedure: place small quantity of test substance on white test plate and add 1 drop of formaldehyde, sulfuric acid tears. Orange red color turn to violet color. Dissolve zero point 0.2 gram, zero point 0.2 mg in 2 ml ethanol. 2 ml add 2 ml water. Add 1 drop sodium chloride TS. Orange red solution changes to blue violet color. Dissolve 0.05 gram in 1 ml water. Add 5 drops of ammonia TS and filter. Acidify filtrate with nitric acid TS and 1 ml silver nitrate TS. A white curdy PPT, a white curdy, a white curdy precipitate soluble in ammonia TS but practically insoluble in nitric acid TS. Discoloration test solution. Ketamine hydrochloride, white hygroscopic crystalline powder, characteristic order. Melting point is 260 degrees Celsius. In this, these are 3 tests. We have to dissolve 0.2 gram in 4 ml water, then chill solution in ice. To that we will add that is potassium carbonate TS with pH indicator paper and allow standing to crystallize and filter, and we have dry crystals in vacuum over phosphorus pentoxide. Melting temperature is about 92 degrees Celsius. Then dissolve the 10 mg in 4 ml water. Add 0.5 ml nitric acid TS and 0.5 ml silver nitrate TS. White curdy PPT. Separate